Good afternoon and welcome to this lecture on single-cell omics. My name is Simon Anders. I'm a physicist by training, but now I'm working in the biology department doing bioinformatics or statistics. And as I've written in the announcement, the idea behind this lecture is to give you a bit of, to make this area of biology, this interface between biology and mathematics sort of known to you as the next generation. It's typical that there is a lot of, that there's many physicists working in this area. And I usually like to claim that what I'm doing is still physics, because in a way, physics is I would say, the art of using math to understand the world or to understand nature. And that is exactly what we'll talk about here. We'll try to use mathematical methods, and what I want to show you is how the mathematical toolbox that you especially learn in physics, but maybe also in mathematics, I hope, is very useful to not only understand the non-living matter, like fundamental physics, but also in many areas of the living matter. And there's a huge demand in biology for researchers who have a mathematical training. And so this is a quite worthwhile field to look into and to work at. And so they conceived this lecture originally specifically for people with a mathematically oriented undergrad, so students of math, physics, computer science. I think we also have a scientific computing master, but on the other hand, there's also quite a few biologists who have maybe more in-depth biology, mathematical expertise than the average biology undergrad. This is quite a spread in my experience. So I imagine, or from the announcement, I imagine that we have a mixture of this. Please come in. So the first thing I would like to find out is where you're all from. Who here is studying physics? 1, 2, 3, 4, 5, 6 physicists. And who's coming from math? 1, 2, 3, 4, 5, 6, 7. Okay. And who's coming from biology? 1, 2, 3. Okay. And what's with the others? Who hasn't marked up? Who's doing something else than this? What are you doing? Computer science. You too? Say again? Scientific computing and. Okay, so she has already made the move of going from the quantitative or computational sciences to the life sciences. And now I will show you a little bit. Now, the first half of what I want to do is, as I said, we will look through different areas of mathematics which are useful. And my idea was a bit to give you a bit of feeling, especially how these things that you've learned in a more abstract thing in your undergrad look once you apply them to a real world problem. The specific thing about biology is noisy. In physics, you're used to things being rather clear-cut. You exactly know how many degrees of freedom you have, and you can account for all of them. And typically, you always look at stuff where the complicated degrees of freedom are frozen out, and you look at small systems of few particles and so on. But of course, you all know that things can get more complicated. In biology, this is always the case. So we will do a lot of coarse graining, taking things not exactly. We will not do anything ab initio, but rather we have a big amount of data and just try to find patterns and structures in that. And for this, we will just go through the garden of math in a way and pick different methods which turn out to be useful. And the single-cell omics that I've chosen here I've chosen for 3 reasons. First, it's what I'm working on. Second, it's something that's currently of great importance in current biological research. And third, it's a really nice showcase how you can use methods from high-dimensional statistics, multivariate calculus, and so on in real life. Come in. Obviously, as we have a couple of, quite a lot of you are not biologists, I need to tell you first a little bit about the biology part of it. And so we won't get into the real math today. Today I will sort of give you a crash course in biology, which will be unfortunately a bit boring for the 3 or 4 biologists in the audience. But at the end, you will hopefully have a short idea, a rough idea of where the whole thing is going, and then we can start with looking for some way of finding some mathematical modeling around this data from the next lecture onwards. The newcomers, where are you from? Math, physics, computer science, biology? All math people. Okay, this is very nice to see that there are so many math people here. When this is the second time I make the course, the first time I basically had only students from the Master of System Biology, so all from the biology side. The reason was that I didn't manage to get this known to you math guys, because there's some issue with the information flow. I still haven't managed to, I got your Dean of Studies and your Studienkommission to accept this as part of the math major, and it should officially appear in the Modulhandbuch. But in practice, certain people in the department don't seem to be able to simply add a page to a PDF and put this online. This is why it's still not in, and why in the end it took announcing it by email. And to be honest, I got quite angry about this fact that it's simply impossible to get anything simply in a lecture catalog. I don't know how you guys deal with this, that you can't simply get a catalog of all your lectures without even always having to notice that it's all outdated. But yeah.
It's in the digital picture, so not the PDF, but there's like an online registry.
Yes, and if I ask your people about where should it appear that you guys find it, why do they always tell me the wrong thing? By now, in the end, somebody told me about muesli, so I put it there, but only 3 days ago. So anyway, that was just a little bit of rant against learning that, okay, other departments are even less well organized than ours. Now let's go back to math. So I see we fill this seminar room completely, okay? That should be enough. And as I said, we have to do a biology primer. As I said, I somehow expected that we'd be much more biologists and less people from the other side. But now I'm glad that I used some time yesterday to write together this primer. So let's go through this. What I've done here is I made a course web page, as you can see here, where the link to this thing is on Moodle. And here this is the course material. And now let's first go through this biology primer. I hope we get through this today so that we can start with the real quantitative stuff next time. So this is a long thing, and I think I just make this big. Yeah, it's sort of my brief recap of what you need to know in order to understand what we are going to do here, which is of course only a tiny little bit of what molecular biology has to offer, but it would be enough for you to understand why math is needed here. So let's start with stuff which you probably all remember from high school, at least I very much hope so. What's DNA? You all know each cell has your full genetic information in the form of a long chain-like polymeric molecule, which is just a long chain of A, C's, G's, and T's, which stands for these 4 nucleotides, or bases as they are often called, adenine, cytosine, guanine, and thymine. I'm sure you remember from high school that these things base pair. DNA is a double helix with always 2 strands sitting next to each other, and they pair up in the sense that if the one helix, which is sort of a staircase going upwards, has this sequence, then the other staircase going downwards has something which we call the complementary sequence, where each A is replaced by a T and each G, and have I written here, each A by a T and each G by a C. I have to fix this typo. We call this the reverse complement, meaning we take a sequence, swap all A's for T's and all C's for G's and vice versa, and reverse the thing. And then what we get is what we would read off the DNA, a sequence, if we took the molecule and just turned it around and read it the other way around. The thing is point symmetric, so actually making a 180 turn of the whole thing gives you still the same molecule, but of course with a reverse complementary sequence. So when somebody says we have sequenced the human genome, what is meant by this is that that each human cell contains, as you know, 22 chromosomes, and each of them plus the sex chromosomes, and each of them is a very long molecule with this sequence. And the sequence, which is typically, this is about the longest chromosome has, I think, 300 million bases. The shortest has 40 million bases. So this is the typical thing. Overall, the human genome has 3 billion bases. So this is a pretty long thing. And you can now readily download the sequence of these 3 billion bases as just one long character string from the internet. And we'll do this later, and we work with this. We call this a reference assembly, which is just one set of is just the human genome. So the sequence of all those 22 chromosomes plus the sex chromosomes, X and Y. And you get it simply as a text file where it says chromosome 1, and then comes 200 million ACGT letters, when it says chromosome 2, and so on until you're at the end. Obviously, you know that we all are not genetically the same, we are different. So if you sequence 2 people, then you wouldn't get the same sequence, but different sequences, but most of the base pairs would be the same. Only short pieces differ from people to people. These are called the polymorphic places. And these polymorphisms, this is what makes us unique and what distinguishes one human being from another one, at least at birth, when later you add your life experiences. The neat thing about DNA is, because it's a double-stranded thing, is that nature can copy it. It can just open the strand so that it has the strand individually. And then an enzyme called a polymerase just builds the second strand using the first as a template. And this way, your DNA, your cell can duplicate its DNA. And so when the cell divides to make 2 daughter cells, each daughter cell can inherit the whole DNA. As you can appreciate here, from a computer science standpoint or an informatics standpoint, the nice thing about this, it's digital information. We're used by the fact that in nature everything is analog, it's continuous. But here we have stuff which lives in a discrete space. And this is quite important, and this is in a way part of a thing which makes life possible, the fact that the core information is in a digital discrete state, which hence can be copied in a manner which is non-lossy. So, you all by now, I hope everybody knows what RNA is, not the least because we all got our mRNA vaccine. The DNA is just sitting in our cell. In order to do something with it, the cell makes copies of short stresses of DNA. And this gets made in a different kind of molecule, which is called RNA. DNA stands for desoxyribonucleic acid, RNA for ribonucleic acid. So as you can see, the D means desoxy. So the RNA seems to have some oxygen atom extra somewhere. I can never remember the biochemistry. The physicists probably will remember that the chemistry, at least in my undergrad, the chemistry course was the one thing we immediately forgot afterwards. So that doesn't get better. So RNA works this way. We have another enzyme, also called a polymerase, which can come to a chromosome, open up the strand somewhere in the middle, sort of push the things apart to make a little bubble, go in there and make a copy from one of the strand in RNA. Then RNA is a single-stranded molecule, so it doesn't have this complementary copy, but just this one copy. It has a slightly different chemical way how the different elements are chained together, but that doesn't need to concern us. And just because it sometimes confuses us if we look at data, whenever you write a T in DNA, you write a U in RNA because the nucleotide has a different chemical name when it's in RNA. So this process of making an RNA copy of a piece of DNA is called transcription, and what we then get is a transcript. So a transcript is a copy of a short piece of your chromosome that is then pushed outside the nucleus and swims through the cell. So I should probably have made some. We have a Yes. So in a typical cell, in the simplest case, the cell has a nucleus. In this nucleus are all your chromosomes. And when the polymerase comes, makes a little copy of this thing, and then this copy is pushed outside, and then it swims around in the cell plasma. So this is sort of the main volume of your cell where most of the big biochemistry happens, and this is the nucleus where the information is stored. I probably have to add to offer you a window bench. And I have to see if we find a bigger thing. So, there's different reason why the cell would make copies of a DNA, and the most important is to make proteins. So a protein is again one of these discrete things, a polymer a heteropolymer where we chain together long chains of individual elements, but our elements are in DNA, our elements were the 4 bases, A, C, G, and T. Now our elements are the 20 amino acids. So each amino acid, make this up. Let's open quickly the Wikipedia page. So these things here on this picture, these are the 20 amino acids, which you see have all 20 different chemical things. Some of them are positively charged, some negatively, some are hydrophilic, some are hydrophobic, etc. So they all behave slightly chemically different. But what they all have in common, they can be linked together with something that's called a peptide bond. So the nice thing about proteins is that you can link them together like pearls on a string to make one long chain of these amino acids, which we then call a peptide or protein. And once you have this chain, it's first a one-dimensional thing. And in that sense, if you go along this peptide backbone, you get its one-dimensional sequence. So you can write down the protein sequence, just writing down the chain of amino acids in this 20 amino acid alphabet. But what also happens is once you have these chains, the chain immediately starts to fold into a complex shape because some of these things are charged and attract each other. Some of these things are repelled by water and hence are pushed inwards, while others are pushed outwards because they are pulled to the water molecules. And you remember this all happens in a watery solution. And in this way, what the peptide sequence gives you is first it just codes for a list of amino acids which are chained together in one long string, but it also codes for a complicated 3-dimensional shape which automatically happens when this peptide chain tries to, as we say, fold itself by finding the energetically minimal structure which is optimal for this thing. So, now, where do we get the sequence of this protein from? And this is where our DNA comes in. Each DNA, in the DNA, there's areas which are called protein coding sequences, where a protein is described by giving the sequence of amino acids. And you see here there's a code. For example, this code here, CAG means histidine, or UCA means serine. So always 3 letters of DNA, 3 bases in the DNA form a codon which codes for one of these 20 amino acids. And the code is redundant. There is several codons which code for the same amino acid. And there is one special code which, I can't find it here, yes, here it is, which is called the stop codon. It marks the end of a protein. So what now happens when the cell thinks that it needs a protein? What happens is the cell finds the genetic information about how the protein has to be built, what the sequence of this protein is. This is somewhere on a chromosome. And in this chromosome, the polymerase comes and makes a copy of this piece as RNA. This copy is then moved outside in the cell plasma where another molecular machine comes, the ribosome, and the ribosome then assembles the protein following the directions on this transcript. by always reading 3 bases, looking for the corresponding amino acid, reading the next 3 bases, finding the next amino acid, and chaining them together so that you get this long chain. And while the chain emerges from the back end of a ribosome, it already starts folding itself, so that in the end it gets this folded shape. And this is sort of the central process, the biologists even like to call it the central dogma of molecular biology, that information flows this way. On the DNA, you have a description of a protein, which then gets transcribed into an RNA molecule, which gets moved out of the nucleus where the ribosome finds it and uses it as a blueprint to assemble a protein. Now the proteins, obviously, because we have so many different building blocks, we can assemble proteins in unlimitedly complex shapes and functionalities. So the proteins are the workhorses which really do the things in your body. They either are structural proteins, which are like Lego building blocks to build up the different parts of your cell and of your body, like the cytoskeleton, which keeps the cells intact and so on, or even more important, where the enzymes, the molecular machines, which can take any kind of chemical molecule and attach an atom or remove an atom or move 2 things together or assemble or disassemble complicated biological objects. No, right, this is not what I'm looking for. So this is what I've written here together. We have these proteins. They are, there's the enzymes and the structural proteins. And the sort of core machinery in your cell is the DNA is the cell produces proteins by following the descriptions on these RNA. In the human body, in the human genome, you find blueprints for about 20,000 different proteins. And the cell always has to select which proteins it needs for specific jobs and then will transcribe them, and the ribosome will assemble these. 20,000 proteins is a lot, so you can build a lot of different machines that way. Yep. So here, if you want later, you can also read all this up, the genetic code I have explained to you. Now, these transcript types are, these transcripts, there are different kinds of them. What I've already explained to you are these protein coding genes, parts in the DNA which describe a protein. And if a transcript is made, an RNA copy of this, it allows the cell to assemble the corresponding protein. These transcripts, they have 3 parts. In the middle is the actual blueprint of the protein using the code that I've just shown you with the codons. And then at the beginning and the end are parts which are not part of the blueprint for the protein. They are called the untranslated region, because I've forgotten this process of taking a transcript and using it to assemble a protein. That's called translation. And we'll use this term often. And the part which is not things is called the untranslated region. And they are important for what we call regulation in biology, because you can imagine once this RNA swims around in the cell, whenever a ribosome sees it, it will start assembling a protein, and you will get more and more proteins. At some point, you have to get rid of a transcript when you have enough of the proteins. So there is other proteins whose job is to degrade RNA which are not used anymore. And for these proteins to notice which RNA they need to degrade, they look at the UTR and they find markers there which tell them, I need to, and there seems to be some complicated logic, like when the cell is in this state and has these needs, then this protein should try to find all transcripts which have this sequence in the UTR and degrade them, so destroy them. And this is something which currently in molecular biology research is one of the core questions, that we try to find out how this, what's called gene regulation works. The part with transcription and translation is clear. Once you have a transcript, you produce the protein. But how does the cell know which transcripts to make? Where in the genome should it transcribe something? How does the cell know? How does the ribosome know whether it should not translate this transcript that it's just seen or not? and how do the cell knows when to remove transcripts, and how does can all these complicated regulatory logic be influeed by things which happen outside the cell, like hormones, signals from other cells, stimuli, needs, whatever. So this runs under the general term gene regulation. The idea is that it's an extremely complicated network of this thing interacting with that thing, and if these two comes together, then this reaction can happen, which causes this other reaction, which causes a third reaction, which then causes some protein to get to the DNA, find a certain place, sit there, wait for a polymerase, get this polymerase here, tell it transcribe starting from here, and we try to find these. We are trying to find different elements of this big network, and the kind of data we look on in this lecture will show you one way of getting there. So, besides from protein coding genes, there's also other genes which are, oh, I forgot to point out on the DNA. If this is a stretch of DNA on the protein, on the somewhere in on your chromosome, then there might be places which are transcribed. So maybe this area is often transcribed so that the ribosome comes along and makes a copy. In this area as well. In between are areas which are never transcribed. These things which are transcribed is what we call the genes. In this stuff we call the intergenic part. So in this part of biology, so the word gene has lots of different definitions, but in this part of biology, the word gene means a region on the chromosome which is frequently or regularly transcribed into RNA. And as you can see, most of the RNA, actually not most, but a large part of the genes on the chromosome, they contain the scripts for protein. They are protein coding genes. But there's also other things which are regulatory signals, help the cells to influence something, send other kinds of signals to it, and not to forget, the ribosome itself contains RNA, which is not a messenger, but it's part of a machine. And that's also something which we produce a lot of. So, there's a lot of different kinds of RNA, but most of the time we will be interested mainly in the messenger RNA. That means the RNA which codes for proteins. This here is the Ensemble Genome Browser. It's one of these typical things that that people use to collect what we know about the human genome. So here, the human chromosomes. You see the longest one is numbered 1, the shortest one is numbered 20. Why is 22 longer than 21? Never noticed, but they are sort of ordered by size. And then there's here X and Y. If I now go into one of these chromosomes, I can look into some random part, like this little part in this chromosome, and in this part, let's see when it loads this component. Come on. This. Do I really have to describe this now instead of, okay. This is done by the EBI in Cambridge. They seem to be underfunded. The server gets slower and slower. So I have to draw it then. So typically, if you have a gene which codes for a protein, when you then look at it, there's something odd. This gene is not completely transcribed, but there is the cell makes a transcript of this long stretch, and then it cuts out most of the places and only keeps that part, that part, that part, and that part, and connects them all together to a short transcript, which we call the mature RNA. And these parts which are removed are called introns. So an odd thing about these genes is that the cell first transcribes a long thing, and then it cuts out about 90% of it and only keeps the rest. Big question about why this is the case. So here you can see this typical thing. Here we see a part of a protein coding gene here, which starts here and ends here. And it has these to 3 exons, so parts which actually get, which actually appear in the final transcript, and these angled lines, these are the introns of the parts which are cut out. So these are all little pieces which I now mention in one big go, so that you have one thing. Some of these will become relevant later, others not. What, for example, we might ask, yeah, let's skip over this. Of course, there might be, depending on different circumstances, sometimes an exon gets included, when the next time it gets not included. So there's also variation about how a gene is actually produced in a transcript, because the so-called splicing mechanism, which cuts out these introns, might produce different kinds of final transcripts from one and the same gene. So one gene has several transcripts, and each transcript contains little pieces called exons. So now, this was sort of a rundown of little bits of biology, which we will need later. Of course, it's only a small little part of what we are talking about here. And what I now want to tell you next is high throughput sequencing. So, because why do we have so much data in biology all of a sudden that it's useful to employ physicists and computer scientists and mathematicians to work with it? And that's for a good deal due to the invention of something called high throughput sequencing. By now, DNA sequencing is something which is commonplace. You all know that, maybe you might know that it was a big thing 20 years ago in the year 2003 when the human genome was fully sequenced. I told you before, you can now download this text file, which just gives you the full sequence of all the 23 human chromosomes, just as a long string of A, C, Gs, and Ts. When the first time such a thing was ready, that was, of course, a big thing, and this was called the Human Genome Project. They worked all the way from 1994 to 2003 to it, and when they had it and celebrated, and people looked at it, okay, now we have a string of A, C, G, and Ts, what do we do with it? And that's maybe what we are looking at ever since then. The first thing, which I should have, I think I mentioned it before, is obviously the first thing is you want to annotate it. You want to know where are the transcribed and the non-transcribed part. So you might have a sequence with all these A, C, G, and Ts, and then you mark off in the sequence some area and say, this is transcribed, this is a gene, then this is intergenic, and this is the next gene. This is something we call a genome annotation, just telling us which parts of this long DNA sequence is actually transcribed, i.e. gets copied into RNA, and which is not. And of course, what's the purpose of all the part which is not transcribed? That's also a question we will come back to. So, in order to sequence the human genome, originally this was a big effort. The human genome has a total of 3 billion bases, and in order to sequence this, you use a process for which Fred Sanger got his Nobel Prize, and hence it's called Sanger sequencing. He invented this end of the 70s, when it took about 8 years till the first commercial sequencing machine came onto the market, which allowed us to just give it a piece of DNA, and then this machine spits out the sequence. Typically, this piece could be something like 500 or 1,000 base pairs long, and you would have to make many copies of this same molecule in order to be able to sequence it. Now, making many copies of a DNA molecule is, luckily, that's very easy. Because as I told you before, the cell does this all the time. Whenever a cell splits, it uses this special molecular machine, the polymerase, to duplicate its DNA. And it does that by just opening the DNA, separating the 2 strands, and then taking one strand and reconstructing what has just been taken away to make a new second strand, because after all the information is there. And in that way, people figured out, I keep forgetting the name of the guy who invented it, but he also got rich and Nobel Prize and so on. I And he actually was clever enough to patent it. And besides a Nobel Prize, and this is, whenever you hear the universities talking about innovation, the thing that they always have in mind is that this idea of making the DNA duplication in the cell, taking this machine out of a cell and putting it in a test tube, that earned I think it was the MIT where it was invented, that earned them hundreds of millions. This is, of course, what every university dreams of, that somebody has this kind of idea which you then can patent. By now, the patent has long expired. This whole so-called polymerase chain reaction, or PCR, is done routinely in every biology lab. All of us had a lot of PCRs being done on our corona samples, but there's also other parts. And the PCR allows you to take a stretch of maybe 500 to a few thousand bases and make trillions, quadrillions of copies with it. Simply because in each cycle it doubles the thing, so if you do 20 cycles, you have an exponential amount of DNA. And then you put it in the Sanger sequencing machine, it spits out the sequence. And the machine usually can sequence a few hundred bases or base pairs in a go. Now, if you always can take a few hundred in a go and you want to get 4 billion in the end, this takes a few years. Or back then it took, as I said, 8 years to get through the whole chromosome. When we were about to finish with this arduous process of always taking a bit of DNA, taking the next bit, taking the next bit, stitching them all together, about when they finished, somebody had a new idea, which made the whole process immensely faster and nicer. And that was the idea of high throughput sequencing, which, as the name says, is a method to sequence lots of stuff at the same time. The idea is that the main problem with Sanger sequencing is you have your DNA and you put it in a little test tube, and then you sequence it. And I don't want to explain now how the sequencing works, even though it's a fascinating idea that Sanger had back there. So take your time and read it up on Wikipedia, how the Sanger sequencing works, because it's really a neat idea. But in the end, what you do is you use fluorescence. You get the thing to light up in 4 different colors corresponding to A, C, G, and T. And if you have a lot of mixture of many different DNA things, you get all these different light colors at the same time, and they all blur together, and you don't know which belongs to which sequence, so you can't read it. This is why you can only have one sequence in each test tube. And then people had the idea, let's do the following. Let's take a big plate, and we have a test tube which contains millions of different DNA fragments. For example, I might have taken a human chromosome and just split it into lots of different pieces which are only a few hundred base pairs long. And then we functionalize the surface such that DNA sticks to it. And then one such piece might stick here, another here, a third one here, and so on. And in this way, several million little DNA molecules will stick to the surface, in each of it somewhere else. And when you run your polymerase chain reaction with an extra trick, which is called bridge PCR, and again, I don't have the time to explain it, but it's also a very neat trick for which somebody, I think, probably also got rich, which causes that all these many duplicates of the DNA, if this one gets duplicated by the PCR, usually if you do this in solution, it would swim away. But on the surface, it immediately sticks to the thing, so that you get 2 to the 10 or so copies here, and 2 to the 10 copies of this, and 2 to the 10 copies of that. And when you do with Sanger sequencing, which, as I told you, causes the different nucleotides to flash up in 4 colors of light in sequence, and you watch with a microscope and see, okay, here red, here green, here blue. So the first base is here A, here C, here G. When you take the next cycle, when this is red, which is blue, and this way you read of all these sequences in parallel. And a few hours later, or 2 days later, you have not one sequence of a few hundred base pairs, but a million sequences of a few hundred base pairs. And this is called high throughput sequencing, and it of course revolutionized the thing, because now it doesn't take a year to sequence a chromosome, but only a week. Or by now a few days. where was I? The trick with this, the other point here was that when this idea came up, there was another issue. People looked at this and said, okay, if we take a human chromosome, we fragment it into lots of small pieces which are a few hundred base pairs long, put it onto this flow cell, sequence all these parts in one go in parallel, and now we need to assemble them. We need to bring them in the right order. And of course, if you have many DNA, many copies of your chromosome, and you fragment it up, maybe one molecule gave you that fragment, and the next molecule gave me that fragment, and they overlap. So you look through your DNA pieces and look for overlapping ends, and then you say we have to stitch them together. So the whole thing is a giant but one-dimensional jigsaw puzzle where you need to puzzle together your pieces. So the idea that this should be possible with so-called shotgun sequencing, where you just make lots of different pieces and then puzzle them together, this idea came up in the 90s and people made quick back of the envelope thing and said, man, not possible to puzzle this together. There are simply too many combinatorial possibilities. And around the time when this high throughput sequencing came up, Moore's law has made computers fast enough that people suddenly realized, actually, with the current computer generation, which was sort of a standard in the early 2000s, this has become possible. And so nowadays, if we assemble genomes, if we sequence a new species, we stitch it together that way. And this is why we call this an assembly, because we assemble the long sequence out of all these small fragments. And in that way, many of the other, many animals and plants have been sequenced. And now you can sequence something like, it only takes a few weeks to sequence a new species. If you already have an assembly, the whole thing gets much easier. I don't have to puzzle this together. So for example, if I now want to find out how does my genome differ from somebody else's genome, I might sequence my DNA, get lots of these pieces, and then I look for these pieces in the big reference and align them to them. So I make a string search where I try to find the string of each read in the existing chromosome sequence. And most of the time, the string will match exactly, and sometimes it will only match with a couple of mismatches, like my string search algorithm says, yes, I can find this string that you give me with 300 base pairs, but I don't find it anywhere. I don't find it, I find it only at one place, and there I have to put it. And it must stem from here, but we have to assume that these few bases are mutated. So this is how now all these things work. If you, for example, try to find genes associated with diseases, you sequence the DNA of all the patients, align them, as we say, to the reference genome and find out where the specific subject differs from the whole, from the reference. Now, the next thing is, let's now talk about sequencing RNA. If you take, you know that all the cells in your body have the same DNA, at least, more or less, except for copying errors. So how comes that you have so many different cell types? You have skin cells, you have muscle cells, you have nerve cells. They all do something different, even though they have the same DNA. And obviously, this is because they use different genes. The skin cell will express the genes for the keratins, the proteins which make up the hard outer shell of your skin. A muscle cell will express the genes for myosin, the molecular motor which allows a muscle to contract. A red blood cell will express the genes for hemoglobin, the protein which carries your blood, your oxygen. So every cell type expresses different genes. And this means that if you want to know what's going on in a specific type of cell, a good strategy might be to simply not sequence the cell's DNA, but the RNA. So we extract all the RNA from a cell. then we can, when we transform the RNA chemically into DNA, so we just make it a reverse transcription, that's possible, and then we sequence as before. And in that, and this is called RNA sequencing or transcriptomics, because we get all the transcripts of our sample. And I show you here how this works. So what we do is we take a tissue sample, and this tissue sample we chemically break up all the cell walls so that all the contents of the cells flows in solution, and then we use other chemical tricks to extract the RNA and translate the RNA back into DNA, and then we sequence that, and then we get from our sequencer this text file with a few hundred millions of short reads, so short pieces of DNA sequence. And now we can take each of these reads and ask, what is this read? So first we make a string search looking for this DNA string in our whole reference genome, so in our known sequence of all the chromosomes of a human or of a mouse. And then once we found a match, we look in the gene annotation which gene this is. And then we might say, okay, this read is from the myoglobin gene, or this read is from the hemoglobin gene, or from the keratin gene. And in the end, we get a table which tells us for each gene how many reads we have found which map to this gene. And then we might want to compare different kinds of samples. For example, we might want to know which genes are expressed in all types of cells and which genes are specific for skin or for muscle or for liver. And in that way, if we do RNA sequencing on different tissues, we can find out what the difference between them is. What's more important is we can also use this to learn something about, say, diseases. Maybe we want to understand let's take as an example, some skin disease works. So we take from dozens of patients with, say, psoriasis, Schuppenflechte, a skin sample from an affected area and a skin sample from healthy skin. And when we sequence all this and always check which genes have more reads in the diseased skin sample than in the healthy sample. And in the end, we might say, okay, the problem in this skin disease might be that the diseased cells produce too much of these genes. We call this searching for differentially differentially regulated genes. So genes which whose expression strength, so the amount of transcripts you find in the cell, is consistently higher in all the samples from diseased tissue than all the samples from healthy tissue. And in that manner, we can find out something which hopefully gives us an idea of how to treat this disease. So this is done a lot, and it's a whole industry, but there's one catch. If I take a typical skin sample, then I might have a problem that if you take skin, it doesn't contain only one type of cells. Typical piece of skin contains keratinocytes, so actual skin cells which make up the top of the skin and which produce the upper layer of your skins. Then other epithelial cells which produce the lower layers. You will find hair follicle cells which make the hairs. You will find even small muscle cells which brighten up the hairs whenever you get cold. You will find endothelial cells which line the small blood vessels, the capillaries in your skin. And if you now find that a certain gene is upregulated in the diseased sample, you might ask, which cells were these which upregulated that? All the cells produce more of this gene, or is it that only the epithelial cells produce more, or the blood cells, or the blood vessel lining cells, or the muscle cells, or whatsoever? So you want to know how this whole thing differs. And now, 5 to 10 years ago, a new technology came up which works the following way. You sequence only a small amount of cells. Before, for this RNA-seq, you have a big tissue sample with maybe millions of cells. Now you sequence just 1,000 cells. You dissociate the cells, so you try to split up the tissues so that the cells aren't attached to each other, but that each cell freely floats in some test tube. And when you find some way of sequencing the RNA of each cell separately, in the easiest case, you use this by just using what they call in the lab a microtiter plate, just a little plate with a few hundred little wells, and you put one cell into each well with a suitable machine for this purpose. And then you sequence them all separately, each as its own little sample. And then in the end, you get what's in these cells. You can distinguish the different cells. Specifically, if you have a read, you know, do these two reads, do they come from the same cell or did they come from different cells? The original idea was with these little wells, and then a big improvement came with the idea of microfluidics. Don't know who knows about microfluidics. That's all this clever engineering idea of making chemical reaction in microscopic little channels, which you somehow etch into a chip. And in that case, you make a little water droplets in oil, and each water droplet contains one cell, or hopefully contains one cell. That doesn't really work, and we come to this later. But the idea is that each water droplet contains one cell, and it hopefully doesn't happen, or rarely happens that more than one cell is in one water droplet. And each water droplet contains a little silicon bead, which is coated with a DNA oligo. Now, a DNA oligo is a little piece of DNA with some random sequence. So in each water droplet, you have a different random sequence. Maybe in one water droplet, the sequence is ACGTCCA, and the second it's something else. And there's a very neat trick how you produce this and how you ensure this. And maybe if we have time, I'll tell you later how this works. But in the end, each cell now is put together with this little silica bead, which contains billions of copies of this one and the same oligo. And with another fancy biochemical trick, you now make sure that each RNA read not only contains the copy of the actual RNA molecule, but attached to it is this little oligo from the bead. And hence, each read contains now first the sequence of the actual RNA molecule, followed by the sequence from the bead, which tells you which bead it was, because each bead has a different one. We call this a barcode. The biologists like to use the word barcode whenever they have some DNA sequence which is essentially random, but which allows us to distinguish different entities. So in that case, we get these reads. In each read, we can do two things. We can first find out which gene is that part from, the RNA part, and then we ask which which bead is that one from. And then we can put together all the reads which have the same barcode and say they came from the same cell because they came from the same droplet. And in the end, what we then get is a big table, a big matrix, where each row corresponds to one of the genes, and each column corresponds to one droplet or one barcode or one cell. And the entries tell us how many reads have been found which map to this gene, that's the row corresponds to, and that have a barcode that is of this cell. And this is the kind of, in this matrix is what we will talk about a lot in this lesson. Now, I managed to give you a crash course through all kinds of biology in less than 1 hour. Now you're probably a bit flattened, which is, it's also late in the day, but what I now want to do is to show you how this data looks like by actually loading such a matrix and having a look at it with you. And then we can see what actually the problem is and why we need some advanced math to deal with it. So, for this, I like to use a programming environment called R. Who in the audience knows about R or has worked with R before? Okay, quite a few people. For the others, it's a programming language, a specialized programming language for statistical data analysis, and it's very popular in the life sciences. And there is a huge amount of packages for all kinds of special circumstances. And what I will now use is a package called Seurat. It's named for the Georges Seurat, was a French painter who lived about 100 years ago. He pioneered a pointing technique which was called the pointillist technique, the pointy things, and you will see in a moment why the single cell field felt reminded of Seurat's painting, because we will produce some data, we will produce a plot consisting lots of little points, like Seurat's paintings, where he always used the brush to only make individual little points. And with Seurat, because over this, as I said, the single cell idea came up maybe 10 years ago and has matured ever since and has now widely used since 5 years. And a lot was, of course, discussed how to best analyze it, and some standard best practices came out. And this Seurat package is just a package which gives us software where we can do such a standard analysis. And you will see in a moment that the way how I will show it today is I use it as a black box. I just take my count matrix, I put it into Seurat, and then comes out a plot. And then you will wonder what happened in this black box. And that is what we'll talk about the rest of the term. And, but first, let's have a look at this count matrix. So let's see. Here is my Seurat object. So for those who don't know R, you will quickly notice how it works. Here I load a couple of packages, the Seurat package and a few others which I need. And now I first load my data. The data that I will use is actually a data set that 10x has produced. So 10x is the company which commercialized this water in oil droplet idea. And they manufacture a machine which they call the, forgotten how they call the machine. Everybody just calls it the 10x machine. And this 10x machine sells quite well. One such machine is standing over there, and it has this microfluidics idea, which puts all these little cells into their individual little droplets. And 10x also manufactures these silica beads, where each bead contains a different random DNA barcode, but many copies of it. And in order to demonstrate their product, they put out a couple of example data sets. That's something that they did long ago, maybe 5 years ago. It was one of the first ones. It only contains 3,000 cells. By now, they can routinely do 20 or 30,000 cells. And it contains cells from the blood. It's called PBMC, which stands for peripheral blood mononuclear cells. But peripheral blood means it's simply blood that is taken from the arm vein of some donor, like a doctor would take blood of you. And now you know your blood contains different kinds of cells, red blood cells, white blood cells, platelets. And mononuclear cells means we only keep those cells which have one nucleus, one such thing, which you might have imagined is the normal case, that each cell contains one nucleus with one copy of DNA. But actually, there's quite a few cells in your body which contain several nuclei, 2 or 3 of them within the same cell. In the blood, with certain types of white blood cells contain several nuclei, and it's rather easy to filter them out. There's also cells which contain no nuclei at all, namely the red blood cells. So saying PBMC is just a biologist's way of saying these are the normal white blood cells which don't have this strange extra nuclei. It is only the white blood cells and not the red ones. So we have from some donor, probably some guy in the 10x office who was just asked, can we quickly draw some blood from you? We got 3,000 of these white blood cells, and we will now see that we can make out that there's different types of white blood cells. You might know that in the immune system we have the adaptive immune system, which uses all your immunity, which you train by vaccinated, and these are the B and T cells. And there's the innate immune system, which reacts immediately to a potential threat, which are the, for example, the monocytes and various other things. Now, I don't want to get into the immunology. I always mess up the cell types anyway. But We will now see in a moment that we can tell for each cell that we sequence which kind of blood cell it is. But we will see that this is not as simple as it sounds. So, and what they've done is they took these 3,000 cells, ran them through their machine, sequenced it, then they made this big table and put this table on their web page, and I downloaded it from their web page, and now I load it here. And there is this strange read 10x command, which reads such a matrix in this specific format that 10x uses for their web page, for their thing. And if, Ctrl Z, and if I now look at this count matrix, you see this is how it looks like. It's a sparse matrix. Sparse in mathematics, the mathematicians will know, at least those who've done numerics, we call a matrix sparse if most of the entries are 0. And in computer science, we have special algorithms which store and use matrices differently if we know that most entries are 0. So instead of just storing the matrix by writing all the numbers row by row onto our hard disk, we only store where there's a non-zero entry. That saves storage and it saves RAM, and it also speeds up calculations because you don't spend all your time calculating 0 by 0 if you multiply two matrices. So this is a sparse matrix, and that's not quite surprising because typical mammalian cell has something like a few 100,000 RNA molecules. And if we take these few hundred, but once you sequence them with this 10x machine, it actually loses most of these molecules. It only manages to catch a few percent of them. If you ask the 10x people, they say something like 15%. If you ask other people, they say something like 5%. So from our 100,000 molecules, we only get down to we only get something like maybe 3,000 molecules, which we actually can sequence for this specific cell. And I told you earlier, the human genome has 20,000 protein coding genes and another 40,000 non-coding genes. So if our matrix contains rows for each gene, it contains something like 50,000 rows. But if each column only sums up to maybe 3,000, you can imagine that most entries are 0. This is why this thing here looks like it does. Here you see each row is a gene, so these are gene symbols, which you can look up what they mean. Or if you're a biologist, you might see some of these abbreviations, say, yes, I know this gene. And the columns here are the barcodes. So these are these random barcodes that each bead has. So I show you a bit. So as you can see, in R has this thing that whenever you have a matrix, it stores 2 extra string vectors, which gives each row in each column a name. And here are all the row names, which are all these different gene symbols, where you find genes which one might know or not know. For most of these genes, you will actually find a Wikipedia page. And if I now look at the column names, I see each column is marked with one of these random barcodes, which was on the silicon bit that was together with the cell in the storeblet. And now if I look at this matrix, for example, the first thing I could do is to calculate the column sums, just to see how many reads do I actually get per cell. You see, for this cell, we have sequenced 2,000 molecules. In that cells, it was 8,000, and in that it was only 1,000. I take the decadic logarithm of this, like this, to get it, because then I can make a histogram. And you can see here now the histogram. So this is log 10. So this means 1,000. This means 10,000. 10 to 3.5 is 3,000. So you see most cells have around 2,000 reads that we got. A few of them have many more. A few have much fewer. We can also maybe have a look at the row sums. I know, let's do it the other way around. So let me have a look at my notes. What did I want to show you? The count matrix. Here, these things, the dimension here, and so on. Histogram, no, where was I? Here, this is the histogram that I've just shown you. Now I've decided I just take one arbitrary cell, let's say cell number 1423, so this column, and if I take that, see, it is just one column of a matrix with lots of zeros. So in order to see what the non-zeros is, let me sort these entries so that we have the interesting ones on top. And then you see here the gene which is most strongly expressed in this cell is called B2M. Then comes the gene called NALAT1 and so on. And as you can see, we have all these numbers, and it goes down quite quickly. Soon there's lots of genes where we have only 1 or 2 counts. And you can already now, if you think a bit about the statistics behind this, notice where a problem might be that we get to. If you remember that from these maybe 100,000 genes, 100,000 molecules, we only sequence 3,000, with a selection process and with a random process. It's actually described by a binomial distribution. So there is also binomial or Poisson variance, or the physicists will call it shot noise or Poisson noise in these numbers. And we will need to get around this shot noise in order to still say something about it. Because, let me now pick 2 cells. For example, these 2 cells here, and just plot them, make a scatter plot of one cell against the other. So this is the first cell, just some random number. What did I take? Cell number 1352 and cell number 700 something. And each point is a gene. And then we see here there's genes like these ones, which have around more than 20 counts in both of them. There's genes like this one, which has 20 counts in one cell, but only 1 count in the other. And this here, I put a little bit of jitter. So this is a whole block. So by jitter, I mean each point jitters a bit around its point so that you can easily see how many points are on top of that. So here you see a huge amount of genes which have 0 in both cells. Notice that I put a +1 here, or which have 1 point in both of them. And you do see some correlation. There is a couple of genes which are high in both. There's a couple of genes which are specific to one cell or to another cell. But in the end, what we will get to in a moment is the question, how can we say whether 2 cells are similar or not? And for this, we have to ask, is this correlation high or low? And this, of course, depends a lot on how much variance here is, because all this noise here, this variance, which goes against the correlation, these are not really differences. This is just Poisson noise, shot noise, because the count is so low. We have 2, 3, 4 sequences here. Now, what do we do with this? I told you there is the Server package, which has a certain standard pipeline of steps which we do, and I will explain all these steps in details in the following lectures. But for now, I just run it through and let this thing here do these different steps, normalize the data, scale it, run a PCA, make a neighborhood graph, and so on. And in the end, what you say is this thing here. This is now the pointillist plot that we get. And this is the main working thing that biologists like to stare at. This is an odd kind of plot. It's terribly misleading. There's a lot of things in this plot which do not seem to it, but first I should explain how it works. So what you see here, we call this a UMAP plot, and UMAP is a dimension reduction method. And one thing that I want to do in the course is that we program such a dimension reduction ourselves to really understand what's going on there. So each point here is a cell, and we will put a metric on our cells, such that for any pair of cells, we can say the distance between these 2 cells is this value. So we put a distance metric so that we can say for any 2 columns in our count matrix how similar they are. And if 2 cells are similar to each other, they are placed close to each other in this plot. and if they are very different, they are placed far apart. The way how this works is actually, roughly speaking, you have a big distance matrix which tells you for each pair of cells their distance, which we still have to talk about how we best define this distance. And then you now have two distances. You have the actual distance in the data, and you have a distance on the screen. And now you want to find an arrangement of a point on the screen such that the actual distances between each pair and the screen distance is as close as possible. And it turns out, don't know any of you who have done a bit of astrophysics, know about galaxy simulations, know these little swirly things that they always do at the MPA, where we have lots of stars, and then you see how they attract each other by gravitation and then form a spiral galaxy. If you want to do this, you calculate a simulation where you take each pair of stars and just calculate what's the gravitational pull of them, and then you add up the way how they move towards each other. And here we do the same. We take each pair of cells and ask if they are closer on the screen than they are in our data matrix. We say they repel each other, and if they are farther apart here than before, then they attract each other. And then we run a bit of a simulation and see which picture the thing converges to. The thing that I've just described is something that people tried out long ago and immediately noticed it doesn't work. There's lots of issues, mostly about that our original dimension, our original data, we compare these huge vectors in this matrix with 50,000 rows. So each vector is in a 50,000 dimensional space. And it's unlikely that we will be able to find a global mapping of this high dimensional space into 2D, which isn't completely distorted. Just think about the globe. If you try to make a two-dimensional map of the Earth, you won't manage to make a map where the distance between 2 points on the map is proportional to the distance on the globe, because you have to cut open the globe and you need to find a map projection. So if you can't map 3D onto 2D, how could we map this high dimensional data into this? But we need to find a compromise which allows us to look at the data, even though we know that it will contain distortions, but we will need tools to minimize these distortions or to warn us where the distortions are. And here you see our first mathematical task that we will discuss in the next weeks. How do we do that? Given a big data matrix, how do we first define distances between cells and define distances in a way that they are not completely messed up by all the shot noise? And second, how do we then find a reasonable compromise to project it onto 2D? The biologists, they don't care about this thing. They just tell us, math people, please do it and give us the plot. So I will quickly tell you what happens next. So what the biologists then do once we give this to them. And they are happily ignorant of all the mathematical difficulties in getting there. What they simply ask is, what can we tell about the biology here? And if you look at this plot, you can see there is 3 big blobs. And each blob is made up of cells which are reasonably similar to each other, but very different to the cells from the other blobs. So obviously, there's 3 different types of cells in our blood. Now, if you take any biology textbook and look up what kind of cells are in the blood, you will learn that there is 3 types of white blood cells with only one nucleus: B cells, T cells, and monocytes. And this is why we get these 3 blobs. What you would then typically do is you would first, our biologist who knows that there's B cells and T cells would of course ask, which are the T cells? The biologist knows that a T cell has a T cell receptor. That's this protein complex on its surface that it uses to feel whether whatever it is whether this other thing that's close to the T cell is a friend or foe. Because you know T cells are immune cells. Their job is to go through, patrol the body and take every object and figure out, is this a foreign substance? Is this maybe a pathogen? Or is this just another body cell? And this feeling thing which touches another cell and checks out whether this other cell is correct or not, whether it contains foreign substance, maybe a virus particle. That's called the T cell receptor. And the T cell receptor is made up out of different proteins, and one of these proteins is called the T cell receptor epsilon chain, and the gene which codes for it is called CD3E. And if we look into our count matrix, we will find that there is a row labeled CD3E. Count matrix, no, here am I. Count matrix CD3E. And as you can see, there is a couple of, let me make a tabulate around this. Tabulate. Table. As you can see, there is lots of cells have 0 or only 1 read for this thing, but a couple of cells have 20, 30, or even 34 reads for this gene. These might be the T cells. And Seurat now has this nice function which just colors the thing by the intensity of the T cell gene. And now we can see, yes, in fact, one of these clusters, namely the top right, these are the T cells. Our immunology biology textbook will also tell us that B cells have a B cell receptor, and the gene coding for it is called CD79A. For a strange reason, now we know these are the B cells. Maybe we don't know what these cells are, and we now ask, what's actually the difference between these cells and the other cells? So for each gene, we ask, is there a statistically significant difference between the mean value of this gene's expression? When I say expression, I mean how many read counts we get from this gene for each cell. We ask, is there a strong difference between the average expression of a gene here and here? And we do this for all our genes. Unfortunately, the thing is a bit low and needs 2 minutes for that. We do this for each row in the count matrix. We compare the average over these cells and the average over those cells. And then the thing will tell us for which rows or for which genes we have a strong difference. And that will then allow us to decide what we do there, to look at these genes. And if we now know our immunology and we know more about blood cells, we will look at these genes and say, yeah, obviously these are monocytes, because among the genes which are much more strongly expressed here than there is that gene which every immunologist knows is a typically monocyte protein. To do this, as you can see here, I told Seurat, please compare the cells in cluster 1,4 with the cells in cluster 0,2,3,5. Look at this here. Cluster 1 and 4 is that one, these two, brown and teal. And cluster 0,2,3,4 are here red, green, blue, and olive. So in order to make it easier to say which cells I mean, Seurat has made this helpful coloring where it always made groups of cells which are similar to each other, and said and asked which of these cells are, and gave them the same color. They call this the clustering. And they define a cluster as such a cell. Now, this clustering is also an interesting algorithm. What we have as an input for the clustering is a is a number of vectors for each of these cells, a vector, and some way of defining the distance between these vectors. And now we want to find groups of cells which have a small vector, which are close to each other. And when single cell sequencing was new, people were wondering how to do that. And there is a couple of clustering algorithms which the biologists like to do. So clustering in here means we have some vector space. And in this vector space, we have a group of a few thousand vectors which we are given as numerical vectors. And we somehow want to find, put these find groups of vectors which are close to each other in this vector spaces metric. And to do that, there's a couple of algorithms which are commonly used in biology, but they all only work if you have up to a few hundred vectors because they scale quadratically, and if you have 10,000 points, they get too slow. So the first bioinformaticians who tried to analyze this data looked around and wondered, where can we find something, an algorithm which allows us to find such clusters in big collections of tens of thousands of vectors. And interestingly, they found that this problem turns out in another area, completely different area, in sociology and marketing. Marketing, for example, you work for Spotify and you want to make an automatic music recommendation system. So what you do is you make a giant matrix. Each column is a song. Each row is a Spotify user. And you put a 1 if this user liked this song. And now if two songs are liked by the same people, they are probably similar. So you might assume that two songs are liked by the same people when they are probably similar. And if there's now one user which has liked one song, then you might recommend the other song to this user. But there's millions of songs in Spotify's database, and we need to find how they fit together. And we need, for a given song, we need to find other songs which are close by. And what they did is, what you will use is something called a nearest neighbor search, which is a standard issue in data analysis. You have a list of vectors, potentially tens of thousands of vectors, and for a given vectors, you want to find the 20 other vectors which are closest in distance to this given vector. And if you look for this, there is exact solutions and approximate solutions. And the most general, the implementation of the approximate solution that everybody uses, and that's also used here in this thing, is funnily enough published by Spotify, which is why I take this thing. So we take an algorithm which was developed to solve a marketing problem of Spotify now to analyze our biological data. And we will talk about how this algorithm works, obviously. But the general idea, what we do here is we make what we call a neighborhood graph. We imagine that each cell is a vertex in a graph, and we draw an edge from each cell to its 20 nearest neighbors, which we have identified using, for example, the software of Spotify. When we have what we call a neighborhood graph, where each vertex is connected to its 20 most similar other vertices. And you can imagine that in such a graph, where is my stuff here? In such a graph, if we have something like this here, maybe these are all the B cells, which are all connected to each other, and these are all the C cells, which are connected to each other, and there's only sparse connections between this and that. So we have here a clique of tightly connected stuffs, and we have here a clique of tightly connected vertices, and between these vertices is only few edges which connect this group of vertices with that group of vertices. And we now, of course, if I draw the graph, this is obvious, but what if you have a graph in your computer just instead of a big edge list which tells for each vertex its neighbors in the graph? How do you find, how you best color the vertices, that vertices of the same color are tightly connected with each other and vertices of different colors are not tightly connected? Again, we find the solution in a very different area. There's a couple of mathematical sociologists which were interested in friendships. And at some point, they got their hand on the big Facebook graph. You can imagine that Facebook has a giant graph where each user is a vertex. And when two users are Facebook friends, you have an edge between them. And now at some point, Facebook made this graph public in an anonymized shape, and a lot of math sociology people took that and tried to do some research on it. And what they did, and they had now exactly the same problem. They assumed that there might be face group cliques, like groups of people who are all friends with each other, and they wanted to find that. And then they came up with something which they call modularity score, where they say, if I have my graph and I color the edges in different colors, then I can see whether this coloring is a good reflection of the actual community structure. And we say, we calculate a score, and whenever we have an edge between two vertices of a good color, this gives a plus point, and whenever we have an edge between two vertices of different colors, this gives a minus point. And now how do you have to weight your points such that you get a useful score? And how do you find the assignment that optimizes this score? So this is what we call, the answer is you use the Louvain algorithm, which was made by the University of Leuven, or Louvain in French. So, and that's what I find fascinating from a mathematical point of view about the single cell field, that we have such a diverse area of mathematics. Just in order to get through this thing, we used graph theory. We used specific numeric for big graphs. We will notice that actually in order to define the distance between two different cells, we will need to do an eigendecomposition of our data. And we will have to think about why an eigendecomposition is the right thing to get rid of a Poisson noise, which is actually what I want to talk about next lecture. But this is sort of now a bit of teaser, a bit of preview for you to see where we're heading at, because we will look at these different things. And said I made this lecture already last year, and last year I basically explained the math behind it. And I prepared the whole thing in LaTeX and showed it a bit, and we made a few simple equations, but in the end the students weren't didn't do much themselves, and you just watch the mathematics. Now in this kind of thing, with numerics, and if you actually want to work in this field, you need to be able to program things. So one idea which I would like to try with you now this term is, let's find one of the simpler problems. Let's try to implement themselves, maybe in Python or R or whatever we do. Maybe I'm not quite sure yet how I do this. We sit together, look at maybe I start programming on the screen and you guys follow me, or you take out your own laptops and we all try to find different steps and then go together. But I wanted to try that. When I made this idea, I have to admit, I expected that we'd be 10 people. Now we are quite a bit more. So let's see how this all works out. My original idea was, well, we sit around a thing and do this. But this was an idea to also fill the exercise slot, because last year it was only 2 SVS, so only 2 hours lecture with no exercise. But for you guys to get a reasonable amount of credit points, the math department told me we need to do a bit more work. So I thought, okay, let's have an exercise class. In this exercise class, we might try to actually program something, which of course means, who has some experience in programming? Okay, that's good. Python? Okay, then let's do the whole thing in Python and see how far we get with that. As you see, in order to quickly show something, I like to use R, because R has this nice way that you can quickly load a matrix, take it on in Python until I have remembered how to use this NumPy indexing, or even worse, the pandas .ilog construction. It always takes me too long. But when we do this in exercises, I will have a big advantage that I don't need to know that, because whenever I say, I can't remember how this works in NumPy, I just leave it to you guys and say, who can remind me? So with combined forces, we should be able to do that. And the starting point might be the next time that we play a bit with the original, with the question of how define our distance matrix. And what I want to show you there is, as you've seen it in our matrix, most entries are 0. And if I now want to define a distance between a pair of columns in this big count matrix, how do I best do that? And it turns out that a good way forward is to first log transform it and then do a principal component analysis. Who's familiar with PCA? Half of it, okay, the rest will. But everybody knows what an eigendecomposition is, I suppose. And yeah, if you have a matrix A, then if you do the eigendecomposition of A transpose times A, you will get the PCA. And for those who've forgotten why this is the case and what this is used for and what might be the point of multiplying our matrix with its transpose and then getting the eigenvectors, that might be a good thing, which we can simply try out together. And my idea would be that we can do two ways. We can either write something and apply it to real data, like this blood data, like this blood cell data. Or another very helpful thing is to write a little bit of example of simulated data. So we will sort of imagine a very simplified cell which lives in a state space of only a few dimensions, but from these few dimensions it gets lots of genes, and all these genes we will add lots of Poisson noise to it, and then we will see whether we manage to get rid of a Poisson noise and get back to the original data by using the PCA as a manner of doing clever averaging. So that was the plan for the next lecture. So far it's only a plan. I have to prepare it. I have some notes from last year, but let's go on a bit more from that point. So now I've talked pretty exactly 90 minutes. You all look pretty tired, so probably that's a good place to stop. But one thing we have to do. In my thing, so if we do this sort of sitting together, I wrote in the program that we do lecture on Mondays and exercise on Wednesdays. To be honest, I think it will rather be that we see how this works out, and maybe it will be a mixture and whatever. But the question is first, so I don't have to ask you whether the Monday slot is good for all of you, because otherwise you wouldn't be here. But how about the Wednesday slot? For whom does this Wednesday slot 10 to 12 work? For whom does it not work? 1, 2, 3, 4. So what's the issue in your case? Okay, then that's probably how we have to do it, because I imagine we could try 11 to 1, but I guess there's more people who don't have time from 12 to 1. Or who can do 11 to 1? Who cannot? Now I'm confused. Let's meet here at 10:15. You can come a bit later when you're finished with your tutorial and see how it goes. If you want to work a bit with the computer, you should bring some laptop, maybe with some Python installed and an IDE of your choice. And maybe also install R. If you haven't, if you don't have it, might be useful to have it. For those who haven't worked with R, you need to install 2 pieces of software: the actual R programming language and this RStudio IDE, which comes from a company, but it's actually there's a free version, which is quite useful. And I'm pretty sure we don't have enough outlets to charge everybody's laptop. So try to come with a fully charged computer. And then let's see what, yeah. Okay. Credit points. Biology is weird in the sense they have very tight curricula, and there's actually not much choice options. So I'm not at all used anymore to this credit point counting. What I had done when I made this lecture, I basically talked with Frau Primo-Schmidt, and she said, yeah, best make 2 lectures and 2 exercises. And then I asked her, so what's credit point? And she said, yeah, 2 lectures and 2 credit points. 2 exercises is usually 6 credit points. I don't know if this is the correct calculation or how this works, I'm also actually not at all clear whether I'm at liberty to decide how many credit points this is. I guess this is more the Dean of Studies. So I guess I should double check with, for math, the Dean of Studies is Mammon, I think, right? I think he sort of said it's okay, but we should ask him again whether you really get 6 credit points for the physicists. Yep. At the moment, this thing is in the Modulhandbuch of the mathematicians, which means that all of you can sort of say, I take this as a math minor, because I think you can all take a Mathe-Nebenfach and add it and get whatever credit points the math department says, which is, as far as understood, 6. It might also be nice to add this to the physics Modulhandbuch. I got very contradicting statements from various people at the physics department on whether this is easy or hard. So I probably, who's Dean of Studies in Physics at the moment? Schaefer, so maybe we should ask him, or maybe I should contact him. But yeah, I guess for all the credit points, I'm hesitant to make any statements, because I think in the end it's the Studienkommission which decides that, or the Studiendekan, which simply have to say, and if it's the case that they say, yeah, all these lectures with 2 + 2 are always 6 credit points, then it's probably 6 credit points. If the scheme of assigning credit points is more complicated, like it depends on how hard it is and how much homework there is, then I have no clue. So, yeah. Let me try to find it out, or ask the Studiendekan yourself if you happen to meet him. Yeah.
But we can still note it and have it somewhere to like,
Yes, sure. You can definitely put it in. It will definitely appear in the transcript, I imagine. If we tell Frau Penninger to add it, then it will appear. I think so. Again, for a definite answer, ask Frau Penninger. But I think that's how it works. And yeah, I guess for you to actually get credits, we need to do some kind of work from your side, be it an exam, be it a little project, a little program you write at home. But I thought, let's first try how this now works and decide in 2 or 3 weeks about how we actually make the final. I think in the announcement I said a bit roughly exam or seminar or homework or whatever, because frankly I don't know yet how I best do that. Other questions? Yeah.
The website it was just mentioned the message.
For mathematics, I don't know. It's now in the master Modulhandbuch for the master. At least I hope so, because I haven't seen the Modulhandbuch in its finalized thing. I only saw the page, and people told me they'll put it in. So I hope it will appear in the master, not in the bachelor, because then other people have a problem. I don't know if as a bachelor you take a master level lecture, you should be fine. But again, you have to ask your Studiendekan about this. Okay, if that's your rule, then it is. But yeah, you have to understand, I can't make any statements about your rules, because you have to ask your people. Because I have my diploma or my master's degree in physics, so I'm a bona fide physicist, but that doesn't make me qualified to say anything about the local physics exam regulation. I can tell you in all details the strangeness, as far as I remember, of the degree and credits regulation of the TU München of 20 years ago, but that's maybe not of interest to anybody. Good, then thanks to you all for coming, and see you on Wednesday. And I've recorded this lecture, so I might upload it somewhere.